Hi everyone, it's Dr. LeBlanc. With activation of the Lac Operon by the catabolite activator protein. Our learning goals for this lecture are to understand that operons can have multiple and overlapping types of regulation, to understand activation versus repression, to understand the role of cyclic AMP as a small effector molecule for the catabolite activator protein, to understand the organization and hierarchy of Lac repressor binding domains in the operator region of the Lac Operon, and to understand the three-dimensional structure of the Lac operator and promoter regions within its Lac repressor and cap bound. This is a diagram showing the catabolite activator protein, cap, bound to DNA. Right away, you should notice that the DNA molecule, when it is bound to the catabolite activator protein, is bent. It is quite bent. Catabolite activator protein also has a molecule of cyclic AMP bound to it. Both of these are necessary for it to bind to its site on the DNA. Here's a space-filling model showing that the catabolite activator protein, again, has helices that are inserted into the major groove of the DNA molecule. The Lac operon is regulated by the catabolite activator protein, which is an activator. So the Lac operon has both repression and activation going on to regulate expression of its genes. Cyclic AMP, the small molecule that binds to the catabolite activator protein, is produced in the cell in response to low glucose levels. Okay. If glucose levels are high, ATP is not converted into cyclic AMP. But when glucose levels drop, cyclic AMP is produced. If you think about it, this is a very good way for the Lac repressor, Lac operon to be regulated by catabolite activator protein. If the cell encounters high levels of glucose in its environment, it would prefer to use glucose as a source of energy over lactose because it is an easier, simpler source for the cell to metabolize. However, if glucose is not around and lactose if it's the sole source, then you would want the cell to metabolize the lactose. So if glucose levels are low, cyclic AMP is produced, which binds the catabolite activator protein and helps to activate the Lac operon. So here's an example here. If you have lactose in the cell, but no glucose, then you're going to have high levels of cyclic AMP. The catabolite activator protein with the cyclic AMP bound will bind to its site upstream of the promoter and operator on the Lac operon. Okay? If there's lactose around, then the Lac repressor will have fallen off the operator, and the absence of the repressor and the presence of the activator result in a very high rate of transcription from the Lac operon. You'll produce lots of beta-galactosidase and permease, and the cell will be able to take advantage of all the lactose in its environment. However, if there is no lactose or glucose around, then you really don't want to produce all the proteins from the Lac operon because there's no lactose to metabolize. So even though the catabolite activator protein is bound to its cap site because they have low glucose levels, the repressor will also be bound and will prevent transcription. It's very low due to the binding of the repressor. Okay, what happens if glucose levels are high? What happens if you have both lactose and glucose? Well, you'll get transcription of the Lac operator because the Lac repressor is fallen off, but you won't have it at very high levels because the catabolite activator protein is inactive. If you have glucose, high levels of glucose, then you have low cyclic AMP. That small activator molecule, the small effector molecule, is not bound to the catabolite activator protein. The catabolite activator protein can't bind to its site. And you'll get some transcription, but not at very high levels. And finally, if you have lots of glucose around and no lactose, then you really shut down the Lac Operon because you really don't want to produce any of the proteins from the Lac Operon. Glucose is your preferred source of energy. The cyclic AMP levels are low. Catabolite activated protein won't bind, and the operator is occupied by the repressor, so you really won't be able to transcribe any of the Lac Operon. Okay, so it turns out that the Lac operator has 3 binding sites for the Lac repressor. Here's a picture of a segment of the E. coli chromosome, and we're going to look at the Lac Operon a little bit more up close and personal here. Here's the I gene for the repressor over here, and here's the Lac Operon that encodes the beta-galactosidase, the permease, and the transacetylase. And if we look carefully here, we can see that there are 1, 2, 3 different operators in the upstream and even within the Z gene itself. Now, the sequences of these different operator regions are not identical. They are similar but not identical, which means that some of these operator regions bind to the repressor better than others. When these operator sites are so far apart and the repressor is bound to it, the intervening DNA has to loop out in order for the repressor to grab onto these very distant sites. So now you can see how the repressor really does work. It keeps the DNA looped around, and RNA polymerase will have a tricky time getting through there. Here are the diagram, the 3 different operator sites in the operator region of the Lac Operon. Here is our LAC-Z gene. Here's the promoter. Here is one of the operators. Here's the second operator region, and here's the third operator region. So although we usually diagram the operator region as just downstream from the promoter region here, where the -10 and the -35 sites are, and RNA polymerase will bind, it's actually broken into 3 different sites. The first one is just downstream from the promoter region. The second one is actually within the LAC-Z gene, and the third one is upstream from the promoter. In order for repression to be achieved, the Lac repressor must bind to 2 of the 3 operator sites. Okay, so this is the level of repression over here in this column on the right. Okay, the level of Lac repression in a Lac plus strain, in a wild type strain. Okay, if you have all 3 of your Lac operator sites intact and functioning, you get a level of repression at about 1300. Okay, that's a high level of repression. Nothing is being made from the Lac Operon. However, if you eliminate site number 2, now your repression is only down to a level of 440. These are arbitrary units. Okay? If you eliminate number 3, okay, you increase the repression a little bit more. Okay? So 1 and 2 are most important. Okay? If you eliminate both 2 and 3, then the level of repression drops way, way down. Right? To 18. Okay. If you only eliminate this operator region here, following the one that's just downstream of the promoter region, then repression is almost completely eliminated. It's actually less than if you eliminate these two sites. So clearly O1 is the most important region for the operator. Okay. So like I said before, if you bind to two of these sites, O1 and O3, the DNA loops around. If you bind to O1 and O2, the DNA loops around. Each repressor dimer binds to one of these operator sites. So here's another diagram. We saw this before. Here's the catabolite activator protein right here. It's bound right in this looped out portion of DNA when the O1 and the O3 operator sites are occupied by the Lac repressor. So what does that do for the cell? It makes it primed and ready to go if the repressor is removed. This is a very interesting website, very informative. It will take you step by step through all the levels of regulation of the Lac Operon. And it also has a quiz, and this is the website down here. I highly encourage you to go through this. It's a nice summary and review. So, to summarize, the Lac Operon is regulated both by repression and activation. The catabolite activator protein, cyclic AMP complex, activates the Lac Operon by recruiting RNA polymerase to the promoter. Cyclic AMP levels are high when glucose levels are low. Thus, the Lac Operon is activated to its highest level when both glucose levels are low and lactose is present in the environment. The Lac repressor is a dimer of dimers or a tetramer, and the operator region has 3 different binding sites for the Lac repressor. It will bind 2 out of the 3 sites simultaneously, each dimer binding to one of the sites. One site remains unoccupied at any given time. There's a hierarchy of repression for the sites. Site O1, the central site, is the most critical, and when the repressor is bound to O1 and O3, the DNA is looped around and the highest level of repression is achieved.